Well, welcome to the Mary HYTKEN DUMONT University, where here we study not only direct Torah, but we also have various departments. And recently we've been focusing on our science department. Next week, this week we're going to be talking about to our liturgy department as well, this Wednesday night, next Wednesday night, Thursday night. But now for our science department. And we were talking, it's a series of the wonders of the world. We had one on the wonders of the outer space and understanding about planets and stars and the vastness of the universe. This week about the wonders of DNA. And then in a date to be announced in November, we have Dr. Sharp will be talking about the wonders of the human brain and learning, which is also an area that Dr. Stan is an expert in as well. The Mary HYTKEN DUMONT University remembers Mary HYTKEN DUMONT, who was a member of our congregation, lived across the street, was a wonderful supporter of our congregation. May her memory be a blessing. The topic of the wonders of the human body and DNA remind us of the prayer of Asher Yatzar, the prayer we say every day as after using the facilities. We say that God created the world, created our bodies with great wisdom. And I mean, the wisdom is so obvious to anyone from ancient times to today, how wondrous our body is. The more we learn about it, the more wondrous it is. I work a little bit as a chaplain with organ donations and the wonders of the liver and all the things it does and how it can regrow and all kinds of wonders. And to focus today on really one of the great secrets that the rabbis can only have imagined, and the DNA is very exciting for us. And it's all part of our religious persona. The Sefer Abatim is a book we've been studying recently. It's a medieval Provençal book, and he says that that the mitzvah that we mentioned this past week about gathering Jews together and reading the Torah and the Haftorah and reading on Shabbos or reading on the sabbatical year, reading about the Torah is not only to know the greatness of God, but also lahavim b'maaseh Hashem, to understand and to deepen the works of God. And to me, I can sit in this lecture, we cannot mention God's name one second, or well, Dr. Stan is welcome to, and still, it's really about the wonders of God. And it should really inspire us as we ponder the creation on this Rosh Hashanah that commemorates the creation of the human being. Very briefly, Dr. Stan Eisen is a retired biology professor, having taught at Christian Brothers University for 41 years. Bravo.
Thank you. Thank you very much.
His teaching repertoire included introductory biology, invertebrate zoology, parasitology, and biology of addiction. He is the husband of Ellen Eisen, and he's been married for 44 years. And he's been a wonderful member of our congregation, a wonderful friend and neighbor as well. So we thank him for joining us today and sharing his wisdom with us today on the wonders of DNA.
Thank you, Rabbi, and thank you all for taking time from your schedule to join me in looking at DNA. And it's a bit of a contrast since 3 weeks ago because the speaker, Ms. Levine, was talking about things that were enormously big. The size of these things, the distances between them are just mind-boggling. And I'm proposing to go in the opposite direction and to look at things that are kind of tiny. And in particular, a study of DNA that is found in all organisms. So I have a number of objectives in this presentation: to examine the structure of DNA, to learn the steps involved in the central dogma of molecular biology, and that is that phrase denotes how DNA codes for the structure of the proteins that make us up. And there are 2 steps involved that we'll get to. Those are called DNA transcription, the conversion of DNA into an RNA molecule that is actually used by the cellular machinery to produce a protein, and the actual mechanism by which that's done is called DNA translation. Then to look at the possible consequences of changes in the DNA molecule, and in particular what are called point mutations, where one particular step in the rung of the spiral staircase of DNA will get changed, and whether that can have consequences or not. And then to look at some of the uses of molecular biology in terms of diagnostic, production of medically important biological products, something about our ancestry, and something about the understanding of genetic diseases. So that's my objective, and hopefully we'll get through this in about 40, 45 minutes to entertain some questions. I want you to consider the following 4 organisms that are not drawn to scale. On the left is Sulfolobus acidocaldearis, which thrives in sulfur-rich boiling water of Yellowstone National Park. You would expect that any water that approaches 80 degrees, 90 degrees centigrade, 180 degrees Fahrenheit would be totally hostile to living environment, and yet we find that there are bacteria that live in these very hot waters that we would consider pretty hostile, and they're called thermophiles. And they are very unusual bacteria. There is Bacillus subtilis, known as hay bacillus or grass bacillus, very common bacterium that is studied in microbiology labs. Paramecium on the third panel is a kind of unicellular organism that biology classes in both high school and in college study. And then Homo sapiens, which is the only extant human being, the Latin being for knowing, and was designated in 1758 by a botanist by the name of Linnaeus. And they all have one thing in common. They all have DNA. And in bacteria, this DNA is circular, whereas in organisms with a nucleus, the DNA is found in discrete structures called chromosomes. So can we actually see DNA? And this is from a website that describes the process by which we can actually visualize DNA. And I'd like to do this demonstration right now. It calls for, I'm at step 1 already, mix 500 mils drinking water with 1 tablespoon of table salt. I've done that. It says, let me lower this a little. No, that's not going to do any good. It says to put 3 tablespoons of this mixture into a separate container. Step 4, gargle the salt water for 1 minute. When they say gargle, it really doesn't mean gargle, it means to swish for 1 minute. So if Miss Ellen Eisen would be so kind as to time me for 1 minute in swishing this salt water. 3, 2, 1, l'chaim.
Seven more seconds. Okay, stop.
Boy, a minute can last a long time, can't it?
That was a long time. Now,
It was a long time because I didn't have the clock out.
that was an acquired taste. So if you don't mind my taking something. So now that there's a suspension of my cells from cheek cells. And I want to chase out some of the bubbles that I formed on the walls. Next step. Gently stir salt water with 1 drop of clear dish soap. For the sake of clarity, I'm going to add 2 drops just to make sure this 1, 2. Gently stir. So the soap breaks down the cell membranes, releasing the DNA, and it's still a relatively clear solution. Then, in a separate container, in a separate cup, mix 100 mils of isopropyl alcohol with blue dye. How many drops? Oh, 3 drops. 1, 2, 3. And tilt the saltwater cup and gently mix 100 mils of isopropyl alcohol. Gently pour the alcohol in to form a layer on top about 2 centimeters thick. Now, what I'm telling you now is that whatever it's relatively, the solution is relatively clear. And what I'm telling you is that any glitter, any stringiness, any cotton candy, any white clumps that form is DNA. So it says to let this sit for 2.5 minutes. I'm going to set my timer for 2.5 minutes. And then proceed. We'll come back to this to see if we can find any DNA. Okay, so in this demonstration, what we're looking at is things that are really small. This is a clump of DNA that shows up in this demonstration. So the structure of DNA was worked out by James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. And this article that appeared in 1953, notice, was only 2 pages long. And the central sentence of it is, we wish to suggest a structure for the salt of deoxyribose nucleic acid. This structure has novel features which are of considerable biological interest. And with that modest statement, everything that has come afterward is directly linked to that. All we know about recombinant DNA technology, all the applications are linked to this particular line. And you'll notice that Mr. Watson and Mr. Crick are standing in front of a model of DNA fashioned much like a child would have an erector set or a Lego set of something to produce this. Only Watson and Crick in a Nobel Prize won the Nobel Prize for this work. Without the discoveries of Maurice Wilkins and Rosalind Franklin, however, Watson and Crick would not have been able to work out the structure of DNA. And what Maurice Wilkins discovered was that the concentration of 2 molecules whose acronyms are A and T, adenine and thymine, were equal. Furthermore, that the concentration of guanine and cytosine were equal. regardless of the source of DNA. And what that meant was somehow they matched up in the structure of the DNA. They faced each other on the two rungs of a DNA molecule. What Rosalind Franklin did was a specialist in X-ray crystallography to look at how the structure of things, of crystals, as X-ray beams were fired into it, to figure out the structure. All right, I'm going to pause here. Now, what I'm seeing is the accumulation of strings, not clumps, but strings in the bottom here. Well, it's going to be hard for you to see. I admit that. I don't see any clumps, but I see the accumulation of strings. That's my DNA. That's my DNA. Okay. And what she found was that the structure of DNA is a spiral staircase, it's a double helix. And so notice that the DNA molecule in both examples are double-stranded like a spiral staircase in which, so it's a spiral staircase. The backbones are alternating deoxyribose, which is a kind of sugar, and phosphate groups. And the rungs of the ladder are formed by adenine and thymine lining up, cytosine and guanine lining up. And you'll notice that the artist had put in faint lines to denote weak attractive forces between each adenine and thymine, each cytosine and guanine. 3 weak attractive forces between each cytosine and guanine, 2 between adenine and thymine, and these weak attractive forces are called hydrogen bonds. For the sake of simplicity, what is often done is to unwind the spiral staircase to make it into a ladder. So here is the spiral staircase, here's the ladder, and you'll notice that the backbones are just a little bit different. They have a 5' here, a 3' at the bottom, 3' at the top, 5' at the bottom, which means that even though these two backbones of a DNA molecule are parallel, they're actually running in opposite directions. How do you describe two lines that are moving in complete opposite directions? That kind of configuration is called anti-parallel. So I want you to imagine that my left hand, let me back up, thumb is 5', pinky is 3', my knuckles are some kind of base, and the fingers are hydrogen bonds. 5', 3', 3', 5'. And so between each adenine and adenine and thymine, there are 2 hydrogen bonds. Between each guanine and cytosine, there are 3 hydrogen bonds. Again, 5', 3', 3', 5'. They're going in opposite directions, and that's anti-parallel. That structure of DNA that I've just described accounts for both the replication of DNA and it's specifying the traits that an organism will have. So you'll notice here is an original DNA structure, DNA molecule with 2 complementary strands. They unwind, the appropriate enzymes line up on both strands and produce a complementary strand for each one that was original, so that we end up now with 2 strands that were identical, where at the very beginning of the process we had only 1. So that explains DNA replication. DNA transcription is the formation of a messenger RNA molecule from a DNA template. And here I have a question. Let's just say you have a computer and you want to compose a letter to someone and you use Microsoft Word. Does the code of Microsoft Word in your hard drive, is that the code that the central processing unit of the computer actually use to compose your letter? And the answer is no. The computer makes a copy, a working copy of the code in the hard drive to put it in the central processing unit, and the central processing unit copy is actually used to compose your letter. Well, by analogy, the DNA is the archival copy and it never leaves the nucleus. What does leave the nucleus to be processed to be translated by the enzyme machinery of the cell is a messenger RNA molecule. And there is something about the RNA molecule, the sequence of bases in the RNA molecule that codes for proteins. So let's take a look at this. This is the central dogma, that DNA is stored in the nucleus. An RNA molecule is formed from it. The RNA molecule leaves the nucleus and enters the cytoplasm of the cell, the general contents of the cell, where the cellular structures responsible for producing proteins actually read the bases to produce the protein, and this is translation. And you'll notice in red that I have one exception, and that is to the central dogma. There is such a thing as RNA retroviruses, where the genetic material that is in an RNA capsule is actually RNA. The genetic material is actually RNA, and so the process is somewhat reversed. All right, so the first step in the production of a DNA molecule, of a protein molecule, is DNA transcription. The production of an RNA molecule whose bases are complementary to the DNA molecule. So the two strands of the DNA molecule unzip, and the RNA polymerase enzyme molecule reads each of the bases that are in the DNA molecule and puts its complement in its place. Where it reads a thymine, it puts an adenine in its place. Where it reads an adenine, it puts a uracil instead of thymine. Where it sees a cytosine, it puts in a guanine in its place. And so the direction of the transcription, if you remember my thumb and pinky, is always in one direction from 5' to 3'. The RNA molecule is then shuttled to a ribosome, which reads the bases 3 at a time. So the genetic code is made up of 64 words, and each word consists of 3 letters, 3 bases. And you'll notice that 61 of them code for, actually 60 of them code for a specific amino acid to be put in its place. Question: how do we know that we're looking at the beginning of a sentence when we're reading a letter? How do we know?
It's capitalized.
It's capitalized. And so, and that way we know that this is the beginning of a sentence. Well, here is the capitalization, the metaphor for the capitalization, that the beginning codon for each protein is AUG, and it codes for a specific amino acid called methionine. And so the ribosome reads this 3 at a time, marches and puts together the amino acid the protein 1 amino acid at a time. How do we know that we reached the end of a sentence? How do we know that there's an end of the sentence somewhere? What signal is there? Anybody?
Period.
There's a period. Well, here we have 3 codons that code for stop, which signals to the enzyme machinery that this is the end of the protein. The ribosome disengages, and now we have a complete protein. All right, let's take a look at mutations. We've looked let's look at the effect of mutations. An example of sickle cell anemia, and it turns out that the University of Tennessee, Memphis, and St. Jude Children's Research Hospital are pioneers in the study of sickle cell anemia. Only one base has changed, and you'll notice there should be, it's finally livening up. Let me get back to this, since we're past, I think we're past that. There should be a glutamic acid in this position. Instead, there's a valine. Notice how a normal red blood cell should look like. It should look concave, like a dumbbell shape and profile. And the reason why that's important is the hemoglobin is in a liquid, like a liquefied jello state. And when in the liquefied jello state, it can bend so that it can unload its oxygen when it passes through capillaries. In red print here, the change in amino acid sequence causes hemoglobin molecules to crystallize. So the liquid jello state turns into a solidified jello state. The red blood cells start to change shape. And so now we have a traffic jam in the capillary, and those cells now have 2 problems. Waste materials are not being carried away, nutrient molecules are not being delivered, and that particular area that is being nourished by this capillary dies. That is why sickle cell anemia is so bad. And it's because of a silent mutation. Notice that the genetic code is redundant. We have 4 different codons that code for the same amino acid. So if there's a mutation from CUU to CUC, that mutation is silent. There's no change, no harm. Okay, it's redundant, which means that some mutations are silent. The person will experience no harm. Some mutations can be disastrous. Okay, now let's take a look at some of the uses of molecular biology. Remember Sulfolobus acidocaldiaris, the bacterium which thrives in sulfur-rich boiling water pools at Yellowstone. It, like its relatives, have enzymes called restriction endonucleases, which break up DNA of attacking viruses. And so these are like scissors. They also have enzymes which will reattach sequences of DNA together, glue, if you will. And so there was a conference, a molecular biology conference in Hawaii in 1972, where molecular biologists were talking about the discovery of this restriction endonucleases and saying, what would happen if we took a desirable gene like insulin and transferred it to bacteria, which we can grow, E. coli, which we can grow very readily. And so we can get E. coli to produce a protein that we could use for medically important purposes. So conceptually, this is real simple. This is simply cut, paste, and copy. Your children, your grandchildren do this in kindergarten and first grade all the time. Cut, paste. Now, the the scissors, in this case, restriction into nucleases, let me back up. How much does a children's pair of scissors cost? Let's say $3. To have a dropper full of this restriction into nuclease costs hundreds of dollars. So the copy scissors is very expensive, but the technology is there and conceptually is very simple. So there are some uses of molecular biology. And so, for example, with diagnostics, let's take a look at this. Tay-Sachs disease is a recessive genetic disease. People can be carriers. If they have one copy of the gene, they are normal, and they can lead normal lives. But they can transmit the disease, and it can be fatal by the age of 2. It can be used as a diagnosis for sickle cell anemia. It also can be used as a diagnostic tool to stage or to characterize tumors that look very similar under the microscope, but can be very different biochemically. So here is an illustration of a cell. All cells have the HER, the HER2 gene, and HER2 is a protein that regulates cell division. Cell division is a very carefully orchestrated dance, which starts off with one cell and gives rise to 2. And you'll notice that there are 3 such proteins. There are just 2 copies of the HER gene. And so this is like a gas pedal that is pressed just a little bit down to provide just enough energy and drive for the cell to undergo its division in a slow, deliberate way that is normal. But there are some cells where the HER gene is replicated. And the net result is that the process of replication is sped up. Try to imagine your ability to control a car when the gas pedal is pressed all the way down to the floor. Your ability to maneuver the car in making careful left turns, right turns, brake, whatever the normal driving is hampered. And so this particular cell on the right is overexpressing the HER gene, is undergoing uncontrolled cell growth because it has no brakes. And so what we find is, so this is the kind of thing that is done for breast cancer cells, tumors that are characterized. And we also find that children at St. Jude Children's Research Hospital undergo a complete genetic sequencing for the same reason, because their tumors may look the same under the microscope, but they may be very different on a molecular basis. In terms of the uses of molecular biology, I mentioned that the production of medically important products like human insulin. In the old days, insulin was extracted, collected from the livers of animals that went to slaughter. There were some small, subtle changes, differences in the sequence of amino acids in the insulin of pigs, horses, cows. And in some sensitive people, it led to an autoimmune disease where the insulin that they were receiving wouldn't work. But human insulin is human insulin. So the idea is to cut the human insulin, extract from bacteria these small circular strands of DNA, and open them up, cut them open, and then glue the insulin gene into the recombinant plasmid, and then reintroduce this recombinant plasmid that has the insulin gene into a bacterium that we can grow in the factory setting. It's now called a transgenic bacteria. Grow in culture, extract the insulin, we now have Humulin. Again, the steps are very simple. Cut, paste, copy. The same thing has been done with the human gene for enhancing the production of red blood cells, erythropoietin. There are now molecular recombinant forms of it called erythrin. Crick is another such genetically engineered drug. So now we can produce medically important products with recombinant DNA technology. Now, we can also use molecular biology to determine ancestry. When I was in college, there were two types of DNA. There was DNA that coded for genes, and there was, quote, junk DNA that had no particular purpose. And then after I graduated from college, this, quote, junk DNA that didn't have any particular known application or, quote, purpose became very useful because it would associate people in time and place with certain mutations. So one question: is there a genetic marker, a sequence of DNA bases that is uniquely associated with Kohanim? And the answer is quite possibly. So let's take a look at some basic observations. The definition of a Jew is someone born of a Jewish mother, and for the sake of discussion, I'm omitting converts. But Kahanut, the status of being a priest, a Kohen, is transmitted by the father, and theoretically indicates a descendant of the original priest Aaron, brother of Moses. What do men have? What genetic trait or attribute do men have that women don't have? They have a Y chromosome. Y chromosomes are associated with males, and they are absent in females. So if there is a genetic marker that is uniquely associated with the Kohanim, it would be on the Y chromosome, which is found among males and not among females. Now, here's some definitions. A haplotype is a combination of closely linked DNA sequences on one chromosome that are often inherited together, which will tie a person or a group of people to a specific time and to a specific place. STRs are short tandem repeats, very short sequences of DNA, such as TATT, thymine, adenine, thymine, thymine, that are found as multiple copies of the Y chromosome and at various locations. So how many copies, where they are is another way of identifying similarities between men. So here we go. In 2009, there was an article that was published, Extended Y chromosome haplotypes resolve multiple and unique lineages of the Jewish priesthood. And from the introduction, we've known this for almost 23 years. More than a decade ago, a characteristic Y chromosome haplotype, a Y chromosome collection of DNA sequence, was found to be associated with the Jewish priesthood, a patrilineal dynasty thought to be founded by the first Jewish priest, the biblical Aaron. The most frequent Kohanim lineage, almost half, is marked by a recently reported mutation in a particular area called P58. Based on genotypes of 12Y short tandem repeats, we identify an extended Kohen modal haplotype on the JP58 background. When did this signature strand of DNA become prevalent? The estimated divergence time of this lineage is based on 17 short tandem repeats of approximately 3,190 years ago. Is the rabbi on? I guess not. How long ago was Aaron alive? Okay, I'm sorry. Question: how long ago, rough estimate, was Aaron alive?
Right. So there's a debate as to whether the Exodus was 2400 BC or 1400 BC or 1200. If it was 1200, then it was approximately 3200 years ago.
But the point is within the range of this divergence of the lineage of this particular sequence of DNA. That's my point. That this particular Kohanim lineage starts at a time that's consistent with when Aaron was alive. These results support the hypothesis of a common origin of Kohan modal haplotype in the Near East well before the dispersion of the Jewish people into separate communities, and indicate that the majority of contemporary Jewish priests descend from a limited number of paternal lineages. Interesting. Second question. So notice just the height of the line tells you that this particular sequence of bases, this particular array of DNA on the Y chromosome, is unusually clustered among Kohanim. Now, I mentioned that there's a background to this particular sequence of DNA that will will tie us to a particular place and time. The origin of this lineage comes from its high frequency in our sample of Bedouins, Yemenis, and Jordanians. Now, you can say lahavdil all you want to say that Jews are different than Bedouins, Yemenis, and Jordanians, but the fact is that this is the same general area of the world that Jews originated. Does anybody remember what tune I was humming when I was swishing the salt water in my mouth? Hint, it was the part of the finale, the fourth movement of Beethoven's Ninth Symphony, where he put into music the poem Alle Menschen seine Brüder, all men are brothers. Again, we can say lahavdil, Jews are different than Bedouins, Yemenis, and Jordanians, but the DNA puts us in the same geographical area. Second, determining ancestry, is there a genetic marker or sequence of DNA bases that is uniquely associated with Jews? And here the data are a little bit messier. That there have been some catastrophic events in Jewish history where the number of Jews has decreased to less than 200,000, less than 200,000 people, leading to what's called the founder effect, that the survivors of this kind of cataclysmic event may be genetically very different than what they originated. And secondly, there has been, quote, a significant admixture of genetic heritage from Southern Europe to the Ashkenazic Jewish background. In other words, as people migrated from our ancestral homeland into Italy, into Turkey, further into Europe, Jews mated and converted the local population so that added genetic variety to Jews. But again, the genetic clustering puts us together in a very, very tight cluster. Nearest neighbor joining analysis robustly supported shared origins of most Jewish population with clearly discernible European, Syrian, North African, Middle Eastern branches. The lesson that it tells me is Alle Menschen seine Brüder again. I'm going to pass this. These studies showed that Jews have a tapestry of shared DNA threads with other Jews and that no one thread is sufficient to define Jewish ancestry. Now, we have this particular issue of is there a genetic basis for a Jew can be genetically charged? How many times, or can be a charged issue? How many times have Jews been accused of being genetically inferior? Second, there is one report which supposedly can identify 90 to 95% of Ashkenazic Jews on the basis of genes from a cellular particle other than the nucleus called mitochondria. When I was in college, one of the statements was all DNA is in the nucleus, no other organelles have DNA. And that changed, that there are at least 2 other cellular particles that have DNA that codes for the proteins that are unique for that particular organelle. And the one that's of greatest interest is mitochondria. Mitochondria is where most of the cellular respiration occurs. When a sperm cell fuses with an egg cell, the sperm cell contributes none of its mitochondria. So the mitochondria you have are from your mother. Okay? So there were some rabbis in Israel who said, oh, all we have to do is look at the DNA of this mitochondria, and we have 90 to 95% confidence that this is an Ashkenazic Jew. The argument falls apart when, let's just say we're dealing with the son or daughter of someone who converted to Judaism and then wants to make Aliyah. Ruth's son would not pass this test because Ruth did not have the genetic signature of Jews. She had the genetic signature of Moab. So the lesson to write home about here is that while we can look at the genetic signature of Kohanim in particular, looking at genetic signatures of Jews in general is much dicier. And that's what the data seems to suggest. Lastly, all identifiable groups of humans have a cluster of genetic diseases because they tend to marry within their groups. So among Ashkenazic Jews, there are certain clusters of, there are certain genes that are found in a higher proportion among Ashkenazic Jews than other identifiable groups. Breast cancer 1 and breast cancer 2 genes are an increased risk for breast, uterine, cervical, and testicular cancers. Canavan disease is a deterioration of white matter in the brain. Cystic fibrosis is a calcium disorder that results in a very thick mucus that usually shortens a person's life. Gaucher disease is a specific enzyme deficiency. Tay-Sachs disease is an enzyme deficiency that destroys neurons in the brain and spinal cord. People can be carriers and lead normal lives, but a person who receives a copy of each gene from both parents is a fatal disease that will kill by the time that the person is 2 years old. And that's why there are now tests among Ashkenazic Jews who want to marry. The prospective groom and bride both donate saliva or take a blood test rather, and the results come back as to whether this couple is compatible. In other words, two individuals that are carriers would be considered incompatible. Among Sephardic Jews, they have their own cluster of genetic diseases. Alpha and beta thalassemia, that is a blood disorder, somewhat like sickle cell anemia. It persists because of the prevalence of malaria in the area that Sephardic Jews were found. Some other conditions that I'm not familiar with, but all of these have genetic bases. Okay, I have said enough. and it's been 50 minutes and a lot of detail. Any questions?
Hi. Yes, I have a question. I put it on the chat. My name is Carolina Melul. I'm coming from Sephardi family. My family is from my dad from Morocco, my mom from Spain. This gene, when you do the DNA, can identify Sephardic Jews from Ashkenaz Jews.
Say that again.
There's any gene or any sequence on DNA, so scientific can identify Jewish from Ashkenaz, from Sphard.
There's been more. To my knowledge, there's been more research done on Ashkenazic genes than Sephardic. The only difference, I may be wrong because I may have been looking at the wrong places.
I'm sorry, stop talking. Yeah.
Go ahead.
I just tested for myself. I test 23andMe and also Ancestry.com. Both could tell me I was Jewish, but none could tell me I was Jewish. I actually identify as Ashkenaz. They put everybody in an Ashkenaz classification.
I personally take those tests with a grain of salt. I'm a little bit skeptical about the ancestry of that, and for the reasons that I suspect that not a whole lot of Sephardic Jews have gone through 23andMe to be sequenced, so you're lumped together with Ashkenazic Jews. That's my theory. I don't know. Although, as an aside, this may or may not be interesting to you. Why is the breast cancer 1 gene have such a high prevalence among Catholic women in Colombia? And the answer is that these women are the descendants of women who were forced to conversion during the Inquisition, or whose families left and became crypto-Jews and emigrated to South America, and they brought the breast cancer gene with them. And so the fact that they have an increased breast cancer 1 gene prevalence indicates that they were originally from Jewish ancestry.
Wow. Okay.
I'm sorry that I can't answer your question anymore.
I know. I just wanted to. I was curious. Thank you so much.
Dr. Stan, if I can ask a question, sort of a theological question. Knowing what you know about DNA specifically and about biology generally, does it make you believe more or less? In other words, you say, well, I can figure it out. I can explain it all. So here it is. This is how you mutate, and this is how it works, and this is how you become a human being, or does it, you say, wow, this is ridiculously complex. Like there's there has to be an intelligent being behind this, or do you say, no, we can now figure out exactly how evolution works through the DNA process, and makes me it leaves the door open to the total atheist even more. What do you think?
I am now reading through Jonathan Sacks' Great Partnership, in which there are two ways to approach this. One is the approach of the late Stephen Jay Gould, where science and faith are like two magisteria, two completely different domains that have no overlap. And Jonathan Sacks' argument is a little bit different, that they are integrated, and that science will tell us how, but science doesn't address the question as to why. And if I ask Charles Darwin and his intellectual descendants why, in terms of giving me meaning to my life, it's pretty shallow. So I have to look elsewhere for personal meaning. Does that help?
Thank you. You're saying that different realms, and right? When you put your biology hat on, you see these processes, and that you see the what. If you want to put your religious hat on, you can ask the why, and comes to religious conclusions. But it's different questions, different answers.
Okay, I'm looking at. Is there
other questions? Let's see.
I saw a documentary where there's lots of pure coanim on an island in the Mediterranean found through DNA study. Have you heard about this? It was talking about how we are ready for the third temple. You may have mentioned this. I came in. Okay, what?
Jerba.
No,
Jerba.
I'm not familiar with that particular piece of research, but I guess we're always ready for the third temple. On some level.
Yes. They say there are a whole bunch of kohanim in Jerba.
Yeah, I don't know.
Yeah, we had a shlicha once from Jerba. She was telling us about it.
Is there a way to identify, to differentiate Sephardic and Ashkenazic Jews? I think that, well, like the way to differentiate them really is one way is by the genetic diseases, which is kind of an unfortunate way to do that. Sephardic Jews across northern Africa, Morocco, Middle East. There was a certain form of malaria that was very prevalent, and it is more lethal than the one in this particular strain is more lethal than the 3 others that infect humans. And it has had such a long association with humans that we've reacted or responded to this disease, terrible disease, with genetic forms, mutant forms of hemoglobin that the malarial parasite finds undigestible. As long as you live in a malarious region, you are ahead of the game with this alternate form of hemoglobin. That explains having the sickle cell trait. It explains thalassemia. But once you remove yourself or found yourself emigrating from an area with malaria to an area where it's malaria-free, then thalassemia and sickle cell anemia have a cost to them. But the traits still persist. I don't see any other questions.
Do you think that the field of using our knowledge of DNA is going to be a real game changer in the near future, or do you feel
in terms of
treatment?
Oh well.
You outlined some.
As I said, every child at St. Jude gets genetically profiled. Every tumor gets genetically profiled. And now we're getting to customized medicine where, for example, with chronic conditions like hypertension or autoimmune diseases, to understand someone's genetic background might be helpful in terms of treatment. But then again, there is some news in our genetic baggage that you may not want to know. Suppose that there is a genetic component to Parkinson's disease. Do you really want to know that you have a 90% chance of developing Parkinsonism when you're a teenager or a young adult? I'm not so sure that is to the advantage of the patient. How can you live life to the fullest when you have in this background that I'm going to be hampered? I suppose this happens with people with Huntington's chorea, which is a dominant. Most deleterious genes are recessive. In other words, if you get one healthy copy from one parent, then you can live a normal life. But Huntington's chorea is different. It is a dominant gene that leads to serious neurological disorder. And there's one particular woman who did some research on Huntington's chorea and why it is concentrated in certain families, only to find out that she herself is positive for the gene. So she is on a race to find out as much as possible before the symptoms start.
Any
Hi.
additional comments?
Yes, I have another question. This is Carolina again. I have a question. It's related to the blood type. Is any blood type that could be safe for Jewish people or not necessarily?
Is there a blood type that's more prevalent among Jewish people than others?
Yes, that's my question.
I don't know. I don't know what the distribution is of the common blood types.
All right, Dr. Stan, any final comments?
Well, it pays to stay healthy. That's one thing. I think what's going to happen is that customized medicine is going to be much more. We're going to find more gene products. We're going to find more genetic testing. I think what we're finding with the absolutely stunning speed that vaccines are being developed for COVID-19 is a testament to people's resourcefulness and how we've gone in molecular biology and understanding molecular biology, that there's something like 300 separate vaccines that are undergoing various phases in such a short period of time. Such is the beneficial power of molecular biology.
Thank you. Well,
Thank
we
you.
appreciate you sharing with us, and it's really our privilege to have scholars in our congregation, in our community, and we thank you, Dr. Stan, for sharing with us. A lot of us felt like we were going back to school, and you're definitely a great professor. Thank you so much.
Thank you.
And we will continue this kind of series. It's a little unconventional at a synagogue that we're doing all this science, but I think it's really part of our, my background is a Torah Umada Yeshiva University. We view it as all part of one big package of understanding God's world. And here at this Rosh Hashanah time, getting ready to Yom Tekilat Masechet, the time of the beginning of creation, or certainly a time that celebrates that. We can certainly celebrate this amazing creation. Kulam v'chokhmah asita, God made them all with great wisdom. So thank you, and thank you all for joining us. And again, just to run down, we have so much going on. Contrary to proper relief, Anshei is strong and moving forward. And we have let's see, we got Wednesday night. I'll be talking about the prayers at 8 o'clock. Every night we have the Daf Yomi after Mincha Marv. Mincha Marv, we're going to do a little slichot after Mincha Marv every night. We zoom in for Shachrit, for Mincha. You can get the show from the morning, the evening. We have, of course, Rosh Hashanah. You have to sign up for that. And next week we'll also have our liturgy department. The cantor will be talking on Wednesday night. I'll be talking about Tshuva Thursday night for a special Tshuva lecture. So thanks for joining us. Thank you, Dr. Stan, and thanks for each one of you for joining us. Thank you.
Thanks, Rabbi.
Okay, we'll talk. Thank you so much. It's really great.